All right, so key features or key events that you need to know about muscle and muscle contraction, okay? First off, we have a neuromuscular junction. That nerve goes really close to the muscle. The action potential goes down the nerve axon and sends, and we're going to talk about the neuromuscular junction, makes that muscle contract. How does it do that? Well, that's what I'm going to show you a close-up of. Okay? You got to understand the role of calcium, the role of acetylcholine, and the role of breaking something that breaks down that acetylcholine. Remember, acetylcholine is a neurotransmitter, which is something called acetylcholine esterase. Or some people shorten it by just saying cholinesterase. Okay? And I'll show you that. But once that action potential tells the muscle what to do, then that's where you have to review all that on your own. But basically, it goes down the sarcolemma on the membrane of the muscle cell, goes down the T-tubules, tells the sarcoplasmic, the sarcoplasmic reticulum to release calcium. The calcium tells the actin to be exposed so that the myosin can bind to it, and then you've got contraction, right? All that wonderful stuff. I'm not going to ask you stuff about A bands and I bands and all that stuff, but you should have a concept in your head of what goes on with that, okay, and the role of calcium. So this is that nerve that goes down, and it gets very, very close to a muscle. Never touches it, but it gets really close. Okay, that closeness over there is called a neuromuscular junction. I'm going to do that for you up here. All right, so I might as well do it now. So what's happening here is this. Okay. This is an axon from our nerve. Okay, so going back to this one over here, just so you can see it, right? I drew a close-up of this, right? So you have this nerve. So there's a cell body. Here's the axon. So an action potential is going to come down here, and then we got down here, right? So we have this action potential that's going to come down here. So let me just label what's going on here, and then we'll talk about the events, because all this is going to make sense once you talk about diseases. This is the axon terminal, the end of the axon. The membrane there is called and everything's labeled up here. This is called the presynaptic membrane. This is going to be the postsynaptic membrane. It's on the muscle. Okay? In between is going to be the synaptic cleft. Okay? Inside the axon terminal, we have these vesicles. Inside the vesicles, we have acetylcholine. It's a neurotransmitter. Okay? On the motor end plate, which is the postsynaptic membrane of a muscle, we are going to have receptors for the acetylcholine. Okay? Now, this is what happens. An action potential, and everything is written on here in the next few slides. I'm going to verbalize it for you and do some animation with my own drawings here. The action potential comes down here. That's a nerve impulse. Action potential comes down. Once it gets to the axon terminal, we have calcium that is floating all around here. And there's calcium channels over here. Once the action potential comes down to this area, it opens up the calcium channels and makes the calcium go inside the axon terminal. The calcium is then going to bind with the vesicles. The vesicles are then going to fuse with this presynaptic membrane. And it's going to release acetylcholine into the synaptic cleft. The acetylcholine is then going to bind to these receptors, acetylcholine receptors. Now, once these get pressed like a button, there's going to be an action potential that's going to spread across the whole muscle. That's going to go down the T tubules, release calcium from the sarcoplasmic reticulum. The calcium is going to expose a certain part of the actin to allow myosin to go on there, and you get contraction. So calcium, as you're seeing, and hopefully you saw with A and P, calcium makes bones and teeth strong, but they're more important in neuromuscular junctions, in contraction. It's so important. That's why we need to always have a good supply of calcium that's in the bloodstream to get to these places. Okay? Does this make sense so far? Now, one other thing. If these buttons are always pressed, then this muscle is always going to be contracted. That's not good. I want to pick up this water over here. My hands are going to do this, right? But I got to stop. I can't keep contracting. I got to relax it a little. So you can't have a sustained contraction. This acetylcholine has to come off. Otherwise, you're going to have a sustained contraction. So we have something else that's floating over here. This is called acetylcholine esterase. It's an enzyme that is going to be able to pick up some of this acetylcholine and break it up into two things. It turns it into choline, and that gets absorbed back into the axon terminal for recycling, to make more acetylcholine for future use. It also breaks up and becomes acetate. It's also known as acetic acid, which is also known as vinegar. Acetate is all over your body. It's ubiquitous. It's all over. So it just goes all over with the rest of the acetate. But the thing that acetylcholine esterase is doing is taking that acetylcholine so that you're not going to have a sustained contraction. Is this clear? You sure? Because once you understand this, the diseases are going to be just gravy for you guys. I'll show you. Because if you have too much, here, if this stays on, if we have it so that the acetylcholine stays on here, are you going to have, and here's the choices. You can either have a flaccid contraction, or flaccid paralysis, meaning like you can't move it, but it's just flaccid, right? Or you can have spastic. Spastic. So if these turn on all the time, are you going to have flaccid or spastic? Spastic. If we have something where we're going to block the release of all this acetylcholine, so that we can't have acetylcholine go in there, that means no acetylcholine will bind here. Are you going to have a flaccid paralysis or spastic? Flaccid. Good. Okay? What if we have it where we can put something there to block those receptors so that the acetylcholine can't get there? Would that be a flaccid paralysis or spastic? Flaccid. You see? It's all the diseases. Trying to emphasize, you know the normal, the abnormal just makes sense to you. It's just a list of diseases, and just memorize which one does what. But the concept is not hard.
So the enzyme's always just like chilling in there?
Correct. It's chilling, but it's chilling, yeah. But it's got to break that up. Otherwise, there could be a disease, which we're not going to go over, that doesn't have that, and you're going to have a sustained contraction. All right, we could block that. We could have an acetylcholine esterase inhibitor, a medication that will block that. That means you're going to put a lot more acetylcholine here. And we'll talk about that. We're okay with this so far? All right. Now, all right. So let me just put it back the way it was. Anybody have any questions on this before we get? I'll leave that up there and we'll get back to it, okay? All right, and everything here, this is step by step. This is all the stuff I just explained to you over there. It's just in words. If you go step by step, 1 through 7, here's acetylcholinesterase. 1 through 7, this picture here, 1, 2, 3, it follows the same thing. Like I said, these are the same pictures, I'm doing the same exact thing I do for A&P1. It's the same thing, it's just a review for you guys, okay?
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
All right, and this is showing you, here's a muscle, and then here's a muscle cell. And inside a muscle cell is also referred to as a muscle fiber. It's a really long cell. Inside there, you're going to have the contractile organelles, which are myofibrils, but that's the actin and myosin. And it's loaded with that stuff. You want to have a lot of contractions going on. Okay? Remember this good old stuff? A bands, I bands, and stuff? I'm not going to ask you about that. I put that up there too scared. Okay? So we're not going to ask you about that. But do understand, here's myosin and this is actin. They need to ride on each other, right? To form a contraction. Okay? And then you got the two tubules. The blue stuff here is the endoplasmic or the sarcoplasmic reticulum. The stuff that's all happening in the muscle itself. Okay? just review that stuff so it'll make more sense to you. Okay, here's a close-up of that too.